Okay, now for the rest of this. And I'll go through it real quick, but also I put a lot of nice words there to try and make sure we've got everything. So this we've covered. Two amino acids in the world are going to get joined together, and we don't care how they get joined together now. It's a ribosome, but that doesn't really matter. And when we join them together, we're going to have to take out an OH from here and an H from there to make a water molecule, which is H2O. And in place of that, we're going to end up with this carbon actually joined up to this nitrogen right in the spot where hydrogen used to be. And so, voila. The R group on here, think of that as the rest of the molecule. This is the part that's going to be different on every amino acid. And so, yeah, depending what amino acid we are, this is going to be a different set of molecule or set of atoms that are going to be attached to the amino acid. These are the names of the amino acids. And so we're going to use these single letter amino acid codes because we're pros, not little babies, right? Of course we are. Yeah. And so if we were to write out a protein like this, MFVFLVLLPLVSS, then we know that that actually stands for, it's just like a real compact way to say that we have a protein which starts with methionine, which is then bonded to phenylalanine, which is then itself bonded to valine, which is then bonded to another phenylalanine and then to a leucine, etc. Valine, leucine, leucine, proline, leucine, valine, serine, serine, and so on. These are the first 13 amino acids of the SARS coronavirus spike, and these are actually the signal peptide. So another way to look at what the heck is a protein. So you got your backbone, and so I think of it as like a charm bracelet. So here's our protein backbone, and here's our protein backbone running down the middle. It's the thing with these blue and orange little dots on it. And like a charm bracelet, we're going to have different side chains hanging off to the side here, and so they're each going to have different functions. Some are going to be big or small. And in the case of a protein, some of these things are going to be positively or negatively charged or hydrophobic or, who knows what. And so here's our backbone, and here's some of our side chains sticking out. And so we got one sticking out to the left and right, right and left and right and left, just like that. And so on all the way down to the bottom of the protein. And that's great. So here are the amino acids again. We've already seen something like this. We know that there are these amino acids, which is great. And we know that they're all joined up. So in this one, I think the other one that you saw, the amino acid was sideways. In this one, the amino acid, the backbone part is up here at the top, and the part that is different on each one is down here at the bottom. So glycine has nothing hanging off, cysteine has a sulfur and a hydrogen, and glutamine has just a whole bunch of stuff. And look at tryptophan, oh my gosh, yeah. It really packed for the journey. Okay, here's one way to look at the spike protein. And so from this, you know that this is a three-dimensional structure. And what we're looking at is the surface of it. So if each atom was kind of inflated to a certain size, we were looking at the edge of the electron cloud around that atom, something like that, then we could say that, yeah, it would be roughly this shape. It's not a thing you're ever going to be able to see with your eyes because they're just too small. They're so much smaller than actual wavelengths of light we can see with our eyes that just doesn't, the physics do not work there. I've color-coded this based on charge. And so the really acidic amino acids are the sort of dark red and orange. The basic amino acids are these sort of lighter orangey colors. And then just base normal amino acids are going to be these yellow to slightly green ones. And the real green ones are going to be very hydrophobic. That's the different amino acids we've got. So that's just one way of looking at it. And so once again, this is our protein. So you know this protein. We've got all those domains. And let's break it down. Let's look at the different domains, see what they do, see what each of these little labels actually means. So label number 1 was a signal peptide. Signal peptides are a bit of the protein that occurs right at the beginning and then gets cut off. And it's like punching a ticket. Remember, it marks this protein for export to the cell surface, or if it's on a virus, it marks it for inclusion in the virus and sending out of the cell. Signal peptides all have 3 regions. The first region is going to be kind of polar, usually. And so you'll have amino acids in there that would be in that polar group. So if you look back a couple slides, there was a whole set of those. In the center, you're going to have hydrophobic amino acids. So these are ones that hate water, and they're actually going to embed themselves in the membrane. So that's what the middle of this is for. And you can see it can be different lengths, just like that. And then at the end, we have the a set of small and sometimes charged amino acids. And these are what actually fits into the groove of signal peptidase, which is the thing that cuts off signal peptides. And so signal peptides will cut right after the amino acid shown here. And so I drew in the SARS coronavirus 2, and I highlighted where the different amino acids are. And so there you go. You got a picture of exactly what is being recognized and cut off. So signal peptidase is there on the gene. It's there when the protein is first made, and even while we are making the protein, this is actually going to get snipped off and it will just get thrown away. But it will mark the protein. The protein will get handed off to some other sort of guides, chaperones if you like, that will take the protein eventually out of the cell. Here's another way to look at the spike protein. So in this one, we've gone for the what they call a ribbon diagram. So we're just looking at the protein backbone here. We're pretending side chains don't exist, which just makes it a little easier to see what's going on. And you can see look at all those alpha helices down at the bottom, and look at all of these beta strands up at the top. So the spike protein is broken down into two regions. The first region is called S1, and S1 does some important stuff, including it binds the receptor. So here you can see that the spike is actually made of 3 copies of the same protein. So they're color-coded. So the brown one comes from a single protein chain, as does the green one, as does the blue one. Of these 3 copies, 2 are the same and 1 is different. So there are receptor binding domains. So this is the receptor binding domain. This is actually the part of the spike that will latch on to the receptor. And they can be locked either up, like this, or they can be locked down. So the green one over here is locked in the down position. And essentially, it's just like it's been clipped down, like if this part right here has been clipped down onto the center. So here's one that's locked down right over here next to it. And there's a blue one that's locked down right over here as well. And so when they're down, they probably can't bind the receptor. When they're up, they can totally bind the receptor. And so one gets to be up at a time, and they probably sort of cycle, so that 2 are down and 1's up all the time, because that's the way, when they solve the structure of this, that's just the way it looks. It's always got 1 up and 2 down. So probably the virus is trying to do this during its actual life. And so together those make up the S1 subunit, which is the first half of S. Everything up to the furin cleavage site. And so shown here, the S1 is this first half, and so here is 1 copy of the S1. So on this one, instead of the ribbon diagram or the hydrophobic space-filling diagram, I've gone for just a space-filling diagram where I color the chains by color. But you can see it's the same basic idea as on the previous slide. Here is the receptor binding domain that is stuck up in the air, and the 2 that are locked down, one's here and one is hiding in behind here. And so these 2 that are down can't do anything, but the one that's up, the actual binding site, is right here. So this is in the same orientation as on the previous slide. So S1 is at the top. S1 is for binding the receptor and then getting out of the way. And once S1 has got out of the way, then S2 can fold up, refold, unfold, stick its fusion peptide, which is right down about here, into the membrane. And then the S2 is actually going to make the virus fuse with the cell. So it's going to make the virus enter the cell. So receptor binding domain is the yellow thing shown at the top. The receptor is shown down here in blue. So this is called ACE2, angiotensin converting enzyme number 2. And this is the protein that SARS coronavirus is trying to find in the world and bind onto. And so you can see that the way it binds, it's got this sort of contact face here, and it's curved a bit like a banana, and it's matching the curve of this part of the protein. And then at the end, it's got this little thing. It's kind of like a hook where it's going to reach around and try and make extra good contact with the protein. So here's the hook coming around and kind of bracketing the end of this alpha helix on the receptor. And so there we go. That's how they interact. Oh, and at the interaction points, at each of these points, the little dotted lines are showing what are called hydrogen bonds, which are just the way that proteins are held together. and the way that proteins stick to other proteins. We have one other type of bond here, which is this phenylalanine, which is hydrophobic, and it's interacting with this methionine, which is also a little bit hydrophobic. This is actually called a pi and dipole bond, but we don't really need to know that. We just know that it's sticking together. And so it's got a long face of contact with all these little points where it can stick together. And so that's why it sticks, and that's why it binds, and that's why people get coronaviruses sometimes. Okay, let's move on. So the next little item was the furin cleavage site. This is furin. And so furin is going to operate in pairs, and the pairs are actually going to pair up to make what we call a homotetramer, which means 4 copies of the same protein working together. Each one of these has an active site, and so the active site is shown here, or here, or here, or down here. These active sites are where the protein that's going to be cut docks. So the little black circle shows this little blue squiggle here. This is the protein that is locked in and is about to be cut. And they just solved the structure this way so we could see exactly where the protein is going in. That's kind of a neat thing to do. So that's the first one, and that's going to do the first activation step. The second one, second cleavage, is shown over here. And so this is a model of TMPRSS2, the protease that does the actual activation cleavage, the more important of the two. And this rainbow-colored thing is the little bit of curly protein, so these are the side chains sticking off, that TMPRSS2 is going to cut. Interesting fun fact that we don't really need, but why not? Why not be interesting, right? So this is ACE2. This is the bent helix at the top that's going to bind. This is TMPRSS2, and it turns out that these two things actually interact with each other. So they're actually found as a complex on the surface of cells. Where you find one, you find both. And so it's probably not coincidence that this virus is binding to this one and then is being cut by this one. There are a lot, by not coincidence, I mean there are a lot of proteases that could cut at that TMPRSS2 cleavage site, like cathepsinL is another one. But TMPRSS2 is found already stuck to ACE2 protein, the receptor. And so if it's right there, why wouldn't the virus use it? It's kind of like that. All right, so next part of it is going to be the fusion peptide. And so this is from one of my PhD students, Entidar Al-Saadi. And so what we've got here are little vesicles. So these are little green phospholipid droplets, and they're hollow inside. And what we're doing is putting on the fusion peptide. And I don't know if you noticed that change. Let's go back here. They start out round, and you want to watch this one. As the fusion peptide goes in, this thing is going to go kind of square and then kind of to a hexagon, then kind of to a pentagon. And eventually, if we leave these fusion peptides on long enough, they just deform the heck out of the membrane. They just sort of crumple these vesicles. Because fusion peptide all by itself will actually just embed in the membrane. And when you just start piling protein into the membrane, then it does this to the membrane. It sort of deforms it, as you might imagine. If you're cramming something into pretty much anything, it's just going to deform the surface. And so there we go. That was a nice little, not quite real time. I think that was actually 30 minutes worth of deforming. But yeah, you can kind of see it that way. And so this is the fusion peptide. We just made the fusion peptide for this one. And this was the MERS coronavirus 1. The SARS coronavirus and SARS coronavirus 2 fusion peptides, pretty much the same thing. They work in the same way. I just don't happen to have a video, so we went with the video. Okay, what next? Well, next, now we're down into the S2 region. And so the S2 region is going to be the dramatic shape change region. So we're going to start out with a spike that's kind of blocky and triangular, kind of like this. We're going to end up with a spike that's like a long, skinny sort of baseball bat, like that, or like a wooden cooking spoon, maybe. And so this is the before, and this is the after. And if you notice that slimming weight loss, it's because we've lost half the protein. So the entire S1 region has just popped off and gone somewhere else. It's not attached anymore. It doesn't need to be there. And so when this happens, it allows this to extend and hook the cell and then bring the cell back down. And so eventually you're going to have the fusion peptide right here in the same membrane as the transmembrane parts of the protein, which are right here. And it's going to be driven by these two things called heptad repeat 1 and heptad repeat 2, which is HR1 and HR2. You can see right up here at the top. Forget about the other domains. You don't need those, but FP is the fusion peptide. You need that one. Okay, so what's a heptad repeat? Well, like I said, it's an alpha helix where one side is going to be hydrophobic and the other side is going to be hydrophilic. Here are a bunch from, the world. And so all of these have hydrophobic amino acids, which they're showing in yellow, and look at them just lined up on one side. And they have hydrophilic amino acids, things that are going to love water, on the other side. So with a protein like this, most of these things are designed to actually float in the membrane. So this part, the hydrophobic part, will be stuck into the membrane, and the hydrophilic part will be sticking out of the membrane where the water is. But in the case of coronavirus, it can't get there. And so you've got these poor, unhappy proteins, and what they do is two of these heptad repeat alpha helices will just pair up so that their yellow parts are stuck to the inside and all of their hydrophilic parts are stuck to the outside. That's what we were just looking at. And so here's the entire model, just if you want to know. So we start out with the entire spike. Then we're going to activate it. We're going to cut it here and here to free up that fusion peptide. Once the fusion peptide is free, then the priming stage is, this is when we cut and when we're going to pick up those protons on the histidine molecules down here in the yellow region. Anyway, when we bind protons to those histidines, they're going to be what we call protonated. They're going to gain a positive charge, and that is going to kind of pop this protein and rearrange it. And it's going to rearrange it with the fusion peptide up here. The fusion peptide was still here down in this view. It just was not shown. But it's going to embed, just like hooking the virus into the cell membrane. Then the next thing that happens is that we're going to reel this in, because this is heptad repeat 1 and 2. And these two things have a hydrophobic side right on the inside, and they're going to try and bring those together. And when they bring them together, so here we are halfway brought together, here we are fully brought together, and boom. It basically brings the fusion peptide down to the same place where the bottom of the virus is. And this is called hemifusion. So we've gone halfway through fusion. So this is the virus membrane, this is the cell membrane at the top. Here we've kind of joined those two membranes together halfway, and then from that point, all the virus does is get to hemifusion and hold on. And by holding on, it forces just the laws of physics to clear this up and drive it to full fusion. And when it does, now everything that was inside the virus, look at that, is now connected to everything that was inside the cell. And so this cell is now infected. That's what spike does. That's why we try to block spike so that it cannot start the infection. Spike starts step 1 of the infection, and if there's no spike binding and it can't go through all these crazy little changes, then you don't have to worry about spike. You don't have to worry about the rest of infection. You're not infected. And so that is the point of both the antibodies and of the some of the antivirals, some of the neutralizing antibodies that we're going to talk about. All right, last couple of things. So the protein itself is going to be decorated, we call it decorated, it has to be fancy, with sugar chains. And so these little colorful circles and squares and triangles and diamonds are just telling you that this is a particular sugar chain, and it's always going to be attached to either a nitrogen, and we call this N-linked, or an oxygen in a serine or threonine amino acid, and so we call that O-linked from the oxygen. The N-linked is attached to a nitrogen on asparagine, which is also N, so that kind of works out both ways. And so you can go through and say, oh, okay, this is a fucose that's attached to the side of a N-acetylglucosamine is what GLUCNAC stands for. And N-acetylgalactosamine, I don't know, is the yellow square, etc. You can go through all of these parts. And so there are many different ways to make a glycan. And oh my gosh, you could go study all this stuff, but we don't need to know it. Here are the glycan structure of the SARS coronavirus 2 spike. So shown here, the pink ones are the most complex and highly processed ones. There are 22 points where we have sugar chains attached. Here are all 22 of them. And there is a real mix. Some of these things get heavily modified. Some of these things in yellow are kind of an intermediate level of modification. And some of these things in green are just at a really primitive stage of modification. And you can follow these little bars here. Imagine each bar is a different type of linkage. And so you can see for every single one of these, even these two, where they're most similar, they're not exactly the same. This one has a little bit of, well, let's see, that particular linkage, and this one's missing. Yeah. So there are many, many ways that glycans can look, and the SARS coronavirus glycans just happen to look like this, exactly like this. And this is the virus's camouflage that I was telling you about. So the virus is being careful to leave the receptor binding domain free, so it can actually grab on to the cell receptor. Here's another receptor binding domain that's actually tucked down at the moment. And the other domains, those N-terminal domains, the exterior of S2, they're all about being covered in sugar so that the immune system can't latch on to the coronavirus. Because these sugars are going to be mostly the same as the sugars you would find on the outsides of any protein on the outside of a cell. And so cells can't react against it because the virus is basically making itself look like a cell. Yeah, little tiny wolf in little tiny sheep's clothing. Okay, next modification. And you can see we're pretty much at the end, thank goodness. So this is called acylation or palmitylation. So palmitylation is the addition of palmitin, which is a fatty acid. Acylation is adding a fatty acid just in general. There's different ones you could add. The coronavirus gets palmitin, and so we can call it palmitin acylation or palmitylation. That's what the pros would call it. And so this is the palmitin right over here, attached to a little group that'll help transfer it. This is the protein, and there's our cysteine amino acid sticking out. And so we're just going to get this linked up, bam, right onto there. And once it happens, this is going to be a membrane anchor point. And shown down here, these are the actual clusters of cysteines. And when you mess with these clusters, this is how much the spike is supposed to work. And yeah, knock out even one of these clusters or fiddle around with it. And no matter what you do, the spike is worse, much worse than it used to be. So yeah, palmitylation is important. You don't need every single one, but you probably need about half of these to have a palmitin attached to them. And here is the place in SARS coronavirus 2 where those two get attached. And yeah, shown there, SARS coronavirus 2 wild type. And shown here are some other coronaviruses. And you can see all the black bars are where the cysteines are. And so they're pretty well conserved in pretty much every coronavirus. And they always have this repeating pattern. About every 3 to 4 amino acids is going to be a cysteine and is going to be palmitin acylated. Okay, last tiny thing. This is that D614 mutation. This is the one way that the virus seems to be changing. We do not know what it means because the receptor binding domain is up here. So it's all the way up there. This change is right down here tucked in this pocket. So this yellow molecule here is this yellow molecule. And there's our little aspartic acid, number 614, sticking out there. So it's buried right in here. And yeah, frankly, I don't know. You tell me what the heck this change is doing. So D614 is turning into a glycine, which we think might make the spike a little bit more stable. So it's less likely to come apart. It's hard to fathom how that could make the spike any better. And I guess it doesn't necessarily mean it is getting better. It could be getting a little bit worse. It could be adapting to us. The yellow and sort of golden things are the sugar attachment points. So these are our 22 glycans on the outside. And yeah, just so you know where D614 is. This is the spike. This is the complete anatomy of the spike we have now covered. So your job, should you choose to accept it, and yeah, basically you don't get a choice, is we're going to meet and we're going to go over this slide. And you're going to tell me every single stinking part. And you're going to answer at least 2 of my questions, and you're going to ask at least 2 questions of either me or somebody else in the chat and whatever, yeah. It won't be super rigid like that. What I'm looking for is engagement, not an exact 2.0 questions and then you go into a coma. So we're gonna understand this big freaking colorful rainbow of a diagram because this is the whole spike. We've got the primary sequence the actual amino acids. We've got the bits of the secondary sequence, so the transmembrane is a helix, so we can see that. We've got bits of the three-dimensional sequence. We've got some of these domains marked right here. We don't have any of the quaternary structure, the sort of the way the three-dimensional parts come together, because we're just looking at one protein. But we do also have all the modifications, so all the points at which this thing gets cut, or this thing gets something added, like a whatever those are. It's like oscillation stuff. I don't know. We talked about it, but you're on your own right here, or palmitoylation down there. So have a go. Have a go at this. Let's see how you do. And then from here, this is going to be kind of the end of the hardcore protein science. We're going to go straight into vaccine science. But this is the thing that you need in order to be able to get there. So thanks very much.